Okay, last part of the review. Okay, I apologize that I need to, this is not brilliantly clear when I copied it, but what I'm looking at here as a bunch of cells, and one of the things I can see is that each of these cells is in a different stage of splitting. And so you can see here, this is all about mitosis, all right? And I'm going to remind you of my little thing. What do puppies need? Puppies need a PMAT. Prophase, metaphase, anaphase, telophase. So prophase would be this one where I'm starting to see the chloroplasts, okay? So prophase, you start off, you can't see the chloroplast. At prophase, you can start to see the chromosomes. My fingers, the chromosomes. Metaphase, meta means middle, so the chromosomes line up in the middle, and you can see here clearly that the chromosomes are all lined up in the middle. So this one here would be metaphase. Anaphase, remember the fishing line? It's like the chromosomes attached to a fishing line, and the fishing line shortens and pulls them to opposite sides. That's what's happening here. So that's anaphase. And the last one, telophase, is when it starts to, you can see here, there is a new cell wall beginning to form. So you've got to know PMAT. And really what you've got to be able to do is put them in order. And the easiest way to do this is, right, so prophase, you've only got one cell. So there's prophase. At the end of telophase, prophase, metaphase, anaphase, telophase, you're going to have two cells, right? And the key bit here is that those two cells are identical, all right? Unlike when we talked about over here, when we talked about meiosis, these cells are not the same as that one. On this one, scoot around, these cells are identical, and these are sometimes called daughter cells, okay? They're daughters of the original one, okay? Daughter cells. Now, where does metaphase fit into the cell cycle? Okay. All right, the cell cycle. First off, the cell cycle always starts right here at the beginning of G1. That's where you start. Be careful to get it right, because this one looks a little different. Sometimes it looks like that. It could be like that. It doesn't matter how they organize it, how they put the cell cycle. It always starts at the beginning of G1. G1 is gap 1, where the cell grows. Okay? Now, the number one most favorite thing to talk about when we talk about the cell cycle is what happens in the S phase. And in the S phase, we start with one strand of DNA in the cell, and it makes two copies of itself. That's called DNA replication. REPLACATION. DNA replication. And because that each strand of the original DNA, half of it ends up in each strand like that, it's called semi-half conservative saved. Semi-conservative DNA replication, because half of the DNA is saved in each of the two new strands. That's what happens during the S phase. Now, once the S phase is done, we got these things here, CP, and then I've got one at the end of G2 as well. CP, those are checkpoints. What is a checkpoint? You want to make sure that your DNA is replicated properly. You want to make sure that your cells are proper, that they're going to work properly. And the reason for that is we want to produce not one, but two healthy cells, right? We want two healthy cells at the end of this, all right? And so we have a kind of a timeout where we say, hold on a minute, let's check everything. All our ATGCs are in the right order. We don't have any mutations. We don't have any complications. And if everything is good, we can carry on. Once in a while, if you remember, if it's too messed up, the cell just self-destructs. If it's something that can be fixed, they may come back over here to the S phase and fix it before they move on. Then you get to mitosis. So there's your PMAT, P-M-A-T. And at the end of it, if we started with one cell here, at the end of it, we should have 1, 2 cells which are identical to the one we started off at. Okay, that's the cell cycle. Remember, the S phase is where DNA replicates. The checkpoints make sure everything's working properly. And then we go to prophase, metaphase, anaphase, and telophase. Okay, remember, meta is in the middle, and then they pull apart. That comes after it. That's anaphase. Okay, moving on to this one. How do things get in and out of cells? All right, I'm going to remind you that this is the cell membrane, the phospholipid bilayer. And we can do one of two things, right? Things can either move from high, let me go turn my light back on here. Okay, things can move from where there's more of it, a high amount of it, to where there's low amount of it. So they're going from high to low, that's going downhill. On the other hand, you can also do this. You can go from where there's a low amount of something to a high amount of something, where you go uphill. If you remember the story about how I broke my coccyx, you remember that when you go uphill, it takes a lot of energy in the form of ATP. It's hard work, okay? But if you go downhill, that's easy, and we say it's passive, okay? Passive means no energy. It's easy, no work done. And I want to remind you how to do this. So if you have a question like this, and we've got these X's and we've got these dots, and we've got the cell membrane here, right? This is the membrane. And I want to know, are these X's going in or out of the cell actively or passively? So this is what I do. Count my X's. 1, 2, 3. I'm going to write 3 there. Come over here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Then I look at my X's, follow the arrows, always follow the arrows. So the X's are going this way. I'm going from 3 to 12. I'm going from a low number to a high number. Low number to a high number. That means I'm going uphill, which means these are active transport. Right? It's using energy. Now, count my dots here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. I've got 12 on that side, and I got 1, 2, 3, 4, 5, 6, 7, 8 on this side. Follow my arrow. It's going this way, isn't it? So these dots are going from a high number to a low number. So they're going from a high number to a low number. They're going downhill, which means they don't need energy. They are passive, okay? And that's the easiest way to work out if it's active transport or passive transport. Passive is usually going to have this word with it, diffusion. Because things that diffuse move from where there's more of them, they spread out to where there's less of them. So osmosis, or water, is water, right? So if you've got a question about osmosis, you always look for something about water in the answer. When we were talking over here about things moving in and out of the cell by osmosis, we're moving from where there's a high amount of water or more water to where there's less water. That's why it's passive, because we're always going downhill with water. Okay, finally, last couple of bits. So all the evidence that we have in terms of evolution points to the fact that life forms have descended from a common ancestor. So, the embryos all looking like the same things in early development. Uh, the pentadactyl limb, the one bone, two bones, and then 1, 2, 3, 4, 5 fingers. When you look at that on a bat and a dog and a whale and a human, they all look the same. So we have what we call homologies, right? Things that have the same origin, homologies. And then we've got the gold standard, though, is when you look at the DNA, the ATGC, the sequence of nitrogen bases in different organisms, you see that they have a lot in common. These are all evidence of common ancestries or common ancestors. And then if you also, the last bit, remember when you're talking about the tree branches? So if I've got A, B, and C, A and B are more closely related to each other than they are to C because they come off the same branch. Okay, last bit. Look at this. I'm looking at this and I know this is an enzyme. It has a very unique and a very specific shape. This is what the enzyme is working on. This is called a substrate. substrate. And in this case, this is being broken up into these things, which are the products. Okay. And this is the enzyme. And you look at the enzyme and you will notice that the enzyme at the beginning of the reaction and the enzyme at the end of the reaction are identical. That's how you spot which thing is the enzyme. It does not change. No change. It's identical because this enzyme can be then reused to do this over and over again. What do enzymes do? They speed up reactions. How do they speed up reactions? They reduce the activation energy. Remember the story about Grandma's pizza? The first one had the big hill and it took a long time. The second pizza delivery guy had little hill, still had a little bit of hill, but it was much quicker. The second guy was the enzyme controlled reaction. And obviously remember, enzymes are proteins. Okay, that is a quick run through on some things we've done a while ago. I'm pretty sure you should remember most of them, but go through your notebooks and check through everything you've done.